So, welcome to our tutorial on using MEMSAT-SVM, a predictor of transmembrane helices. So, if you're interested in using this method with the CyPRED workbench, you would come to our CyPRED workbench webpage here. You would select your data input type, which in this case is going to be protein sequence data, so that's already selected. And then you would come down to the predictive methods and we would select MEMSAT-SVM. I'm just unticking the CyPRED method here. Scrolling down, we will need to input a protein sequence, and as per our other tutorials, I've got this sequence at UniProt. It's the bacteria rhodopsin from Halobacterium salinarum, and I'll just cut and paste that sequence into the protein sequence box. We need to give it a job name, so I'm just going to call it MEMSAT test, and then we need to provide an email, so I'm just going to provide my email here at UCL. We now just click the submit button. So this is a somewhat, takes a little time, this prediction, so let's come back after this cut. So after a little time, you should eventually get your MEMSAT-SVM results. And so as with all of our sequence feature predictors, we present you first with the sequence plot, which shows your sequence annotated with the predictions we've made. In the case of MEMSAT-SVM, you'll note that the orange residues here are those residues that we predict to be extracellular. The grey residues are the ones that we predict to be a transmembrane helix. And the white residues are those residues we predict to be cytoplasmic. Coming down the page, we have a panel for the schematic diagrams that the predictor produces. So these are plots that are produced by the methods, were you to say run it yourself on your own computer or so forth. The first panel shows you various features that are calculated along the length of the sequence, and these are combined to make the prediction. These are combined by the support vector machine that predicts this. Somewhat importantly, I think you have the little cartoon here at the top, which just shows you which regions of the sequence are extracellular, those in orange, transmembrane helices in grey, and cytoplasmic regions in white. So if we scroll up to the sequence plot, you'll see that these colours are in concordance with one another. You can also see where MEMSAT-SVM has predicted that a helix might be pore lining, and so this dark blue colour that is added to 3 of the helices indicates pore lining. Scrolling down to the bottom, we also have a little cartoon that re-represents this information. So this grey bar, this grey panel through the centre here is the membrane of the cell, and then the chain, these little rectangles that are inserted into the membrane represent the helices, and we can follow it along to see that residues 23 to 43 is the first helix, residue 57 to 75 is the next helix, and so on and so forth. We have these little flags that indicate which are the pore lining helices. So those are the main pictorial outputs from MEMSAT-SVM. Coming back up to the top of the page, the downloads panel, as always, we have a link for a zip file. So this just gathers together all of the different download files, puts them in a zip file for your convenience. We can also download the job configuration. So this is a CSV file that contains information about the different steps that were used to generate this prediction. And then we can also download the text file. So this is the output of MEMSAT-SVM itself. As I'm running Chrome, I need to click the keep button. We can then read this in any plain text editor of our choice. I'm just going to open Visual Studio Code because I quite like it. So let me just turn off some things and then let's open a file. So I'm going to open the CSV file that we just downloaded. And so this just explains what software was run at each step of the prediction. So step 1 here, we ran SciBlast against UniRef90 to find a number of evolutionary relatives to the input sequence. And then in step 2, we run the actual MEMSAT-SVM predictor itself. So this is the support vector machines that made the different predictions. Opening up the output file. So this is the MEMSAT output file. Let me just scroll back up to the top. This is kind of quite dense. It's kind of initially telling you the various bits of the putative helices it's identified, and then it's kind of scoring them to decide which are plausible helices or not. We can kind of scroll all the way down to the bottom here and get a kind of summary of that information, so the summary of the topology analysis. And here we see each helix that was considered and its final score. And then there are some considered helices that have very strongly negative scores. These are ones that MEMSAT-SVM does not consider plausible. So these top ones are the plausible putative helices, and then this information is used to make the final prediction. In this case, 7 of those helices have been accepted as putative membrane spanning helices, and this topology string just describes those. So it describes which residue positions each of these helices can be found at. So 23 to 43, 57 to 75, so on. No reentrant helices were discovered in this case, but 3 poor lining helices were discovered. So the 94 to 110, 118 to 204, and these numbers, these coordinates should be in concordance with some of the helices in the topology string. We get a little summary. The helix count is 7, and the average score is 14.7 in this case. So let me just close that up. So what should we make of this result? So I know that our test sequence was the bacteria rhodopsin, and that's a sequence that I know happens to have 7 helices. So in this case, in interpreting my results, because I have some prior knowledge about this protein that I submitted, this looks like a pretty strong result. It's always important to corroborate your results with other information you might know about the sequence. And that concludes our MEMSAT-SVM tutorial.